Hello everyone, my name is Manika Majumdar and I am working as an assistant professor for the Department of Food Nutrition and Dietetics at Milagros College. Today I am going to talk about cell structure and its function. It is estimated that there are more than 10 million living species exist on Earth. All the living organisms are made up of cells. When living organisms are made up of single cells, then they are called as unicellular organisms, and when made up of many cells, then they are called as multicellular organisms. All living organisms classified on the basis of cell structure into two groups, that is eukaryotes and prokaryotes. Prokaryotes are like bacteria and eukaryotes are like plants, animal and fungi. Now what are these eukaryotic and prokaryotic cells? All these cells share four key components. These are plasma membrane, which is an outer covering of the cell, cytoplasm, which consists of gel-like cytosol, DNA, which is the genetic material of a cell, and ribosomes are the molecular machines that synthesize proteins. Despite these similarities, prokaryotes and eukaryotes differ in a number of important ways. Now please have a look at this picture of prokaryotic and eukaryotic cells. As you can see, these prokaryotic cells are very simple and very small, whereas eukaryotic cells, they are very complex in structure. Prokaryotic cells, there is a genetic material which lied naked in the cytoplasm, whereas in eukaryotic cells, there is a well-defined nucleus. That means the genetic material is covered by a nuclear membrane. In prokaryotic cells, there is no membrane-bound organelles present in the cytoplasm, whereas in eukaryotic cells, there are many membrane-bound organelles like mitochondria, Golgi apparatus, endoplasmic reticulum, etc. The ribosomes which is present in the prokaryotic cell is 70S, whereas the ribosomes which exist in eukaryotic cell is 80S. In prokaryotic cells, there is a tough, rigid layer is present outside of the plasma membrane that is called as cell wall. But whereas in case of eukaryotic cells, some of the eukaryotic cells like plant cells and fungal cells, they have cell wall, but rest of the eukaryotic cells like animal cells, they do not have cell wall. Now at first, let's see some history about the cell. The term cell was first introduced by Robert Hooke. Robert Hooke is the scientist who first observed the dead cork cell. Later on, the Anton van Leeuwenhoek first observed a free living cells. Now what is the cell? The cell is a structural and functional unit of living matter and is capable of carrying in the processes of life independently. Now as you can see, this is the typical animal eukaryotic cells. In a eukaryotic cells, it is called a protoplasm, and this protoplasm is a living part of a cell which is surrounded by a membrane called as plasma membrane. Now this protoplasm is divided into two major organizations. One is nucleus, and surrounding the nucleus is the cytoplasm. Now in this nucleus, it composed of nucleoplasm, genetic material, nucleolus, etc. And this nucleus function is to control the activity inside a cell. Cytoplasm, which is present surrounding the nucleus where various cellular organelles are embedded, which carries out the activities of a cell. Now, at first we will talk about cytoplasm. Cytoplasm, it is a part of a protoplasm which surround the nucleus, and it is bounded periphery by a cell membrane. This cytoplasm is distinguishable into cytoplasmic matrix and cytoplasmic organelles. So, what is cytoplasmic matrix? It is the clear fluid part of the cytoplasm, and it exists in two states, that is sol and gel. The sol part, it exists at the periphery of the cell, like near to the plasma membrane. It is called as ectoplast, and the gel part, it is present near to the nucleus, that is called as endoplast. Cytoplasmic matrix constitutes 90% of water and 10% of minerals, sugar, amino acids, tRNA, vitamins, proteins, enzymes, etc. Let's move on to cytoplasmic organelle. There are two main types of organelles present in the cytoplasm. One is membranous organelle and another one is non-membranous organelles. Membranous organelles are like endoplasmic reticulum, Golgi apparatus, mitochondria and lysosome, and non-membranous organelle is ribosomes. Along with that, in animals eukaryotic cells, there are centrosome and various fibril filaments and tubules are present. First, we will take up the plasma membrane. Plasma membrane is also called as plasmalemma or cell membrane. It is the outer covering of the cell, and it is a very flexible, responsive and dynamic structure. They are selectively permeable, means they can allow certain ions or nutrients across the membrane. It also takes part in the maintenance of internal environment by active transport of ion and nutrient. If we talk about the thickness of this membrane, the thickness is around 80 Armstrong, and the composition of the membrane is mainly lipid and proteins. Now, this is a ultrastructure of a membrane, that is biological membrane. This is the lipid, and as you can see, there are two layers of lipids exist in the membrane. So, the lipids which is mainly present in the membrane is called phospholipid. So, as there are two layers of phospholipids, so they are called as phospholipid bilayer. Along with lipids, there exist proteins. There are many types of protein exist. As you can see, some proteins, they traverse the whole membrane, and there are some proteins which is present on the surface of the membrane. Along with that, there are glycoproteins and some carbohydrate which is present in the outer surface of the membrane. Now, as you can see, it's a part of a plasma membrane. Here, two layers of phospholipid are present. So, that's why it's called as phospholipid bilayer. If we take one phospholipid, this phospholipid, it has two regions, hydrophobic tail and hydrophilic head. Now, a phospholipid is a lipid made up of glycerol, two fatty acid tail and a phosphate link head groups, and membrane involves two layers of phospholipid with their tails pointing inwards and an arrangement is called phospholipid bilayer. As they have two regions, hydrophilic as well as hydrophobic, so they are considered as an amphipathic molecule. The hydrophilic portion of a phospholipid is its head, which contains a negative charge phosphate group as well as an additional small group which may also be charged or polar. The hydrophilic head of phospholipid in a membrane bilayer face outward, contacting the aqueous fluid both inside as well as outside of the cell. Since water is the polar molecule, it is readily form electrostatic interactions and hydrogen bonds with the phospholipid head. Now let's talk about hydrophobic tail. Hydrophobic means water-fearing. It's the part of phospholipid consists of its long non-polar fatty acid tail. Now this fatty acid tail can easily interact with the other non-polar molecules, but they interact poorly with water. Because of this, it is more energetically favorable for the phospholipid to tuck their fatty acid tails away in the interior of the membrane, where they are shielded from the surrounding water. So that's all about phospholipid bilayers. Now, as you can see, there are some cyclic fused ring structure present in the core of the membrane. These are cholesterols. Cholesterol is another lipid composed of 4 fused carbon rings, and it is found alongside phospholipid in the core of the membrane. Membrane proteins. As you can see, there are various types of membrane protein exist. The main two types of membrane proteins are integral membrane protein and the peripheral membrane protein. Integral membrane proteins accounts for 70% of the total membrane protein, and it cannot be extracted from the membrane without disturbing the membrane. Extrinsic protein or peripheral membrane proteins, they are attached to the phospholipid head and non-covalently to the transmembrane protein. It can be separated by mild treatment. As you can see, there are some carbohydrates present in the plasma membrane, which is always present on the outer surface of the membrane. Carbohydrate group which is present on the outer surface of the plasma membrane, when they are attached to the protein, then they form glycoproteins. When this carbohydrate attached with the lipids, then they are called as glycolipids. The proportion of protein, lipids and carbohydrate in the plasma membrane vary between different type of cells. Like for a typical human cell, protein account for about 50% of the composition by mass, lipid account for 40%, and the remaining 10% come from carbohydrates. One of the important function of the protein is it acts as a transport protein. So, in the biological membrane, there are mainly two types of proteins exist, that is channel proteins and carrier proteins. So, as you can see in this picture, this is a channel proteins. This channel protein function it allows the free movement of ions and water molecules. So, as we all know, the core of the membrane is hydrophobic. So, water and any polar molecules, they cannot be easily diffused across the membrane. So, they need proteins where it is surrounded by a hydrophilic molecule, so that water can easily cross through this channel. Another one is carrier proteins. In case of carrier proteins, the protein needs to bind the ion first or the molecule first to be transported. So, when it comes to transport, there are two types of transport takes place across the membrane. First is diffusion and second one is active transport. Now, what is diffusion? Diffusion is the random molecular movement of the substance through intermolecular space in the membrane or in combination with carrier proteins. Now, this continual movement of molecules with one another in liquids or gases. Now, this diffusion can be of two types. First is simple diffusion and second one is facilitated diffusion. So, as you can see here, this is an example of a simple diffusion. Diffusion is always takes place from higher solute concentration to the low solute concentration, and same thing is happening here in the biological membrane. This is initially at the extracellular fluid, the concentration of this solute is more. Later on, after diffusion, the movement of ion, during diffusion, the movement of ion is taking place from high solute concentration to the lower. So, this process is called as simple diffusion, where they can easily cross the lipid membrane. Facilitated diffusions means they require some kind of protein channels with the help of which the ions or any kind of nutrients or polar molecules can move from extracellular fluid to the intracellular fluid. Now, we will see in detail. Simple diffusion, it means the kinetic movement of molecules or ions through a membrane without any interactions with the carrier protein in the membrane. And the movement of molecules from high concentrations to low concentration is taking place without a protein. That's called as simple diffusion. So, here without any carrier or channel protein, the molecules are moving across the membrane from higher concentration to a lower concentration. So, without using any carrier protein or channels, when the movement of ion and nutrient taking place, that is called as simple diffusion. Now, this diffusion's rate can be determined by various ways. The amount of substances, velocity of kinetic motions, number and size of opening in the membrane. As you can see here, these are the gases, molecules, carbon dioxide, nitrogen and oxygen. They can easily diffused across the membrane because they have got a low molecular weight. Small uncharged polar molecules like ethanol, they can also diffused across the membrane. Whereas water is the polar molecules and lipid membrane is the non-polar. So, it cannot cross the membrane. Large uncharged polar molecules like glucose, there are some ions like potassium, magnesium and calcium, and charged polar molecules like amino acid, ATP, they cannot cross the membrane directly. So, they require some carrier protein or channel protein to cross the membrane. Now, simple diffusions across the membrane can occur by two ways. It can directly diffuse across the membrane, or it can cross the membrane through a channel protein. Let us move on to the facilitated diffusion. Facilitated diffusions means it requires interaction of the carrier proteins. The carrier protein aids passage of the molecules or ions through the membrane by binding chemically with them and shuttling them through the membrane in this form. Now, as you can see here, this is an example of a facilitated diffusion. Now, here the molecules or ions, they are transported from a high concentration to a low concentration through a channel protein. Okay, so this is the channel protein by which the ions or molecules are transported, and this is the carrier proteins through which the ions or molecules are transported. Now, in case of carrier protein, the ion must first bind to the protein and then only it can transport it into the other side of the membrane. Next is active transport. When a cell membrane moves molecules or ions uphill, that is against the concentration gradient, by utilizing energy, that process is called active transport. So, as you can see, this is a membrane, and here the green color box, this is a, suppose this is a solute. At this side the concentration is more, and at another side the concentration is less. So, in case of active transport, the movement of ion takes place from low concentration of solute to the high concentration of solute, and this is possible with the help of energy. When they utilized energy, so here ATP is the molecules which gives energy for this process. So, mainly sodium, potassium, calcium, iron, hydrogen, chloride ions are actively transported across the membrane. There are two types of active transport on the basis of the source of energy used to transport. First is primary active transport, and the next one is secondary active transport. Now, what is primary active transport? In this case, the energy is derived directly from the breakdown of ATP, that is adenosine triphosphate, or some other high energy phosphate compound. So, here is an example of sodium and potassium pump. As you can see, sodium is transported outside, and this transport is taking place against the concentration gradient, that is from low concentration of sodium to high concentration of sodium. Same way, the potassium is going from outside to inside. So, potassium is also moving from high concentration of potassium to low concentration of potassium. So, this uphill movement of the sodium and potassium ions requires ATP molecules. They requires energy for this movement, and this energy they can derive directly from ATP. Secondary active transport. This kind of transport driven by an ion concentration gradient established by a primary active transport system to transport a solute uphill. Now, this kind of transport doesn't require ATP directly. So, they require ATP indirectly. So, as you can see in this example, here the glucose is transported actively from low concentrations to high concentration. So, we already discussed that in active transport it requires energy, but in case of secondary active transport it do not require ATP directly. It takes the energy, it uses the energy indirectly. So, as you can see, in this case it requires ATP indirectly to maintain the sodium gradient. So, with this note I am going to end this lecture. Thank you.